Hi students, good morning. Today we can discuss a new topic, that is genetic disorders. Genetic disorders are generally classified into Mendelian disorders and chromosomal disorders. Mendelian disorders are due to mutation of a single gene. Mutation, you know, it is the sudden heritable changes, and it may be dominant or recessive. And we can easily trace this type of disorders in a family by analyzing the pedigree chart. Okay, and here it follows the same principle of Mendelian inheritance. In this way, it is transmitted from parents to next generation. And the major examples of Mendelian disorders are Haemophilia, color blindness, sickle cell anemia, thalassemia, phenylketonuria, etc. And these disorders are either sex-linked recessive disorder or autosome-linked recessive disorder. First we can study sex-linked recessive disorder, and the first one is Haemophilia. You know, Haemophilia is a sex-linked recessive disorder, and there are many proteins which involved in the coagulation or clotting of blood, and one of such proteins get affected in this disorder. As a result, non-stop bleeding will occur. Even from a small cut, the blood continuously oozes out. Blood cannot clot. Okay, and normally it is transmitted from carrier unaffected female to her son. Females are very rarely affected because they have 2 X chromosome, and males have only 1 X chromosome. And the female become hemophilic only when the mother is carrier and father is affected. But that condition is very rare because up to that stage of life they cannot live. It will not survive. Okay. I will write a cross here. Suppose the mother is carrier. Okay. So this is an X-linked inheritance. This is mother is carrier, and here the father is normal, and the gametes formed like this. When it is combined, what are the progenies we obtain? Here X, Y, here X, H, X, and this is X, H, Y. It is normal female, normal male, but here it is carrier female. 1 X is affected, but 1 X is normal. It is a carrier unaffected female. But what about this? It is affected male. Okay, that is the case. That means the carrier female can transmit the disease to her son. And when the female become a hemophilic, it is in this condition. Okay, the mother is carrier and father is affected. In this case, these two will combine and affected female will produce. But this condition is very rare, I told, because up to this stage they cannot live. Okay, so this is the case of transmission of hemophilia. Clear? Now we can move to our next sex-linked recessive disorder, that is color blindness. It also follows the same pattern of inheritance. It is also X-linked recessive disorder. Here the affected people are not able to distinguish red and green color, and the chances of male being color blind is 8%, whereas the female is only 0.4%. Because the same reason, females have a pair of X chromosomes, but males have only 1 X chromosome. Here also the transmission is from carrier mother to the son. Okay, and the female become colorblind only when the mother is carrier and the father is affected. Okay, these two disorders are sex-linked recessive inheritance. Now, we can study the next type, that is autosome-linked recessive disorder. The first one is sickle cell anemia. Here in this sickle cell anemia, I told it is an autosome-linked recessive disorder. The gene that control is HBA. Okay, if this HBA occur in homozygous dominant condition, it become normal individual. And if it occurs in recessive condition, HBS, homozygous recessive condition, it become affected. That means become sickle cell anemic. And if it is in heterozygous condition, it is carrier. Okay, that is the case of sickle cell anemia. And the disease will transmit only when both the partners are carriers. Then only the disorder will be transmitted. Okay, and here the problem is, the hemoglobin, it consists of 4 chains, 2 alpha chains and 2 beta chain. The problem here is with the beta globin chain. And I will write a part of the amino acid sequence here. Here the first amino acid, one is valine, then the next one is histidine, then it is leucine, then it is threonine, then it is proline, then glutamic acid, then glutamic acid. Okay, 1, 2, 3, 4, 5, 6, 7. I show a part of the beta globin chain, the amino acid sequence. Look at the 6th position. It is the amino acid is glutamic acid. And the base pair which synthesize this glutamic acid is the DNA sequence, that is GAG. This is the base sequence. The complementary strand of it is CTC. Okay, and when it is transcribed into mRNA, it becomes GAG. And this GAG translates into glutamic acid. That is the normal case. Okay, so the 6th position, glutamic acid is there. But when it is affected, when it is sickle cell anemic, what's the change happening here? There is a single base change. It is an example of point mutation. Instead of here in the normal, it is GAG. But here, suppose if the gene combination, this base sequence become GTG, then how the complementary strand? It become CAC. And when it is transcribed into mRNA, it become G. There is no T in RNA. There is no thymine. Instead of thymine, uracil is there. So it become GUG. This GUG, this genetic code determines another amino acid, not glutamic acid. It synthesizes valine. So, in the sickle cell anemia, all other up to this 5, it is okay. It is the same, valine, histidine, leucine, threonine, proline. But in the 6th position, instead of glutamic acid, valine is substituted. It is because of a single base. So, it is an example of point mutation also. Clear? In this way, the sickle cell anemia generally occur. This is the case. Instead of glutamic acid in the 6th position of beta globin chain, valine is substituted. As a result, the biconcave disc-shaped RBC become sickle-shaped. So, its oxygen carrying capacity will be reduced. This is about the sickle cell anemia. That is an example of autosome linked recessive disorder. Clear? Next disorder is thalassemia. It is another autosome linked recessive inheritance. Okay. Here also the offsprings become thalassemic when both the partners are unaffected carriers. The problem is with reduced synthesis of globin chain. Okay. And there are two types of thalassemia based on the type of globin chain it is affected. If it is affecting alpha globin chain, then it is known as alpha thalassemia. And if it is affecting beta chain, then it is called beta thalassemia. Okay. And here the alpha thalassemia, it is controlled by two closely linked genes. These are HBA1 and HBA2. These two closely linked genes control the alpha thalassemia. These are situated on chromosome 16. Okay. And here the problem is sometimes one gene or sometimes all the 4 genes may be affected. The more the gene get affected, the less globin chain will be synthesized. Okay. And in the case of beta thalassemia, it is because of a single gene, that is HBB. And this gene is present on chromosome number 11. Here either the single gene or both the genes are affected. And in both the cases, the problem is reduced synthesis of globin chain. That means it is a quantitative problem because globin chain is synthesized in less amount. But we already discussed sickle cell anemia. There also the globin chain is affected, but the problem is qualitative because not properly functioning globin chain is synthesized in sickle cell anemia. So it is a qualitative problem. But here in thalassemia, it is a quantitative problem. Okay. That is about thalassemia. It is an autosome linked recessive inheritance. And the next one is phenylketonuria. It is an inborn error of metabolism and it is also an autosome linked recessive inheritance. Here an enzyme is required, phenylalanine hydroxylase. If that gene is get affected, what will happen? The enzyme will not be synthesized. As a result, phenylalanine cannot be converted into tyrosine. So, this phenylalanine changes into another form, that is phenylpyruvic acid and its derivatives. These things get accumulated in the brain and that causes mental retardation. And sometimes it is eliminated out through urine also. Okay, that is the case of phenylketonuria. These are all about the different types of Mendelian disorders. We can study the chromosomal disorders in the next session. Thank you.